ammonia, I told you extensively about glutamate and glutamine yesterday. One part is asparagine, I am just telling you one line, and today's class will be basically on urea synthesis, that is urea cycle. So, asparagine synthetase is just like oxaloacetate to aspartate, what I have told yesterday. Similarly, arginine synthetase causes aspartate to asparagine by a similar mechanism called asparagine synthetase. Okay. When the term is synthase, at that time there is no ATP is required. When the enzyme is synthetase, T is there, that means there is requirement of ATP for energy, that you remember. Synthase does not require in general any energy, but synthetase requires energy. So, you see here the enzyme name is synthetase, and that is why ATP is used. Now, urea cycle and its disorder today's class. Basically, you see excretory form of nitrogen are in basically in three forms. One is ammonia, another is urea, and third one is uric acid. Usually fishes, they excrete ammonia as such. So, they are called ammonotellic organism, ammonotellic. Birds, they excrete this extra amount of nitrogen as uric acid. So, they are called uricotelic, and mammals and also we human beings excrete ammonia as urea, that is why it is called urotelic, nothing more than that. So, if you remove ammonia from the amino acid, it is deaminated. Now, the leftover part is carbon skeleton, as I told you. So, that carbon skeleton basically, note down this one only, names. It basically funnel into 7 major metabolic products. These metabolic products you can see here, all the names whatever I have written here, you see these are all amino acids. So, all 20 amino acids funnel into basically 7 major intermediates, and from where the amino acids are also synthesized. Now, note down, one is alpha ketoglutarate, another is succinyl-CoA, fumarate, oxaloacetate, pyruvate, acetyl-CoA, acid-acetyl-CoA. These are the 7 major metabolites which are used made up from the carbon skeleton. So, everything is reused. Only excess amount of nitrogen what is not required are excreted as urea. So, if you consume less amount of protein, so less amount of urea will be produced and less will be excreted. The blood ammonia level will also be low. Similarly, if you take a high protein diet, that means more urea production will be there and blood levels will be high, but it will come down to normal in a normal individual because the excess amount of urea will be excreted in the urine. So, what is this? Can anybody tell what is this? Huh? Krebs cycle. Do you agree? Huh? Krebs cycle. Okay. Krebs cycle. And today we will discuss about urea cycle. Okay. So, you see this is in the circular form. So, why it is not called Krebs circle, but it is called a cycle, the circular 7. So, it could have been said TCA circle or urea circle. Instead of that, it is called cycle. You just think of it like glycolytic pathway. It could have been path, it could have been a way, but the name is pathway. Have you ever thought all these things? No. So my purpose in this class is a great quote from Einstein. Say, education is not the learning of facts, but training of the mind to think. So you start questioning why it is like, then only you will understand. I mean, one thing I am telling you out of context, aims and objectives we had to write in MD thesis. I took 6 months to understand what is the difference between the two, whether they are the same meaning or not. I have inquired from many faculty. They are still not clear what is the aim and objective, and that is the biggest problem. You cannot move forward. So you have to know the meaning of each and everything in detail. Then only you will internalize what is the purpose. Aims and objectives, are they synonymous or are they different? If so, give an example. Okay, you think of it. So nitrogen removal from the amino acids are removal of amino group. Then this transport of amino group to the liver, I have told extensively yesterday in the form of 3 things basically: glutamate, glutamine and alanine. Now this nitrogen enters into the mitochondria, and this prepare the nitrogen to enter into urea cycle, and urea cycle operates. So basically it occurs within the mitochondria. So urea cycle is also called Krebs-Henseleit cycle because they discovered it. এই ছবি তোলা বন্ধ করো, 1000 বার বলেছি লেখো, লেখা একটাই কারণে দরকার, why? Because you have to write in the examination hall. All these things you take it after from me, these are for you. Nothing great. Only thing is that I don't want this thing to be, circulated by WhatsApp. It's a common practice because some things which I have prepared with my efforts, you can understand these slides are not available in the internet. So I have put some extra effort to make this, and I want that you people should have this information. You can come at any point of time, note down. Only issue. So it was the first metabolic cycle discovered in 1932. So urea cycle converts ammonia into urea, and the production site is liver. I have been repeatedly telling the production site of urea is liver, and excretion site is in kidney. Don't confuse it. Subcellular organelle, basically in urea cycle there are 5 steps. 5 enzymes are involved, 5 enzyme defects are there. In addition, there is one more defect, that is a transporter defect. So only 5 steps are there, 5 enzymes are involved, and 5 enzymes are subjected to defects. So they all lead to urea cycle disorder. In addition to that, one transporter is there. So total 6 diseases are basically inborn errors of metabolism under urea cycle or urea cycle genetic defect. So what happens basically, so you see the cell is there, they occur within the mitochondria. Mitochondria means mitochondrial matrix. Not inner membrane, not outer membrane, but within the matrix. Now, third, fourth, and fifth, these occur in cytosol. But the reaction is like this. One again, I should say 1, 2, then 3, you can say, then 4, and 5, and this 5 again participate into 2. Okay. Now I will tell you in detail. Now you see what I have written here is this one. First step. I will make it more visible. You can see these two steps are there, first and second. This is within the mitochondria. Note down here, ornithine is there, it is a substrate. It is not the product. Product is citrulline. But citrulline is shown here, ornithine is shown here, which is a substrate, it is not a product. So, first product is carbamoyl phosphate, that is shown in the first reaction. The second reaction production, carbamoyl phosphate reacts with ornithine to produce citrulline, that is the second one. But you look at this thing, this ornithine is here or not here. Now, citrulline to argininosuccinate, argininosuccinate to arginine to urea. So, each step I am now covering. Now, this is now more visible. Okay. Now, you see urea is for basic formula, if you see, it is CO NA. That means it is having 2 nitrogen. Now, where from these 2 nitrogen is coming? Number 1, that is a big question. Who contributes and where from C is coming? C basically you see, look at the first stage, NH3 plus CO2. So, that CO2 is produced within the mitochondria during Krebs cycle. If you remember, there are 2 carbon dioxides are produced. So, this is readily available within the mitochondria and ammonia is transported. So, that ammonia contributes 1 nitrogen. I have made it, I mean, as a red round. This contributes the first nitrogen. Out of 2 nitrogen, 1 nitrogen is contributed by the ammonia itself. Okay. So, in this reaction, what happens? Carbamoyl phosphate synthetase I is required, this enzyme. Carbamoyl phosphate synthetase I, and as I tell you that this is a T is there, so ATP will be required. So, in this reaction 2 ATPs are required and 2 ATPs, 1 ATP acts as a phosphate donor for the mixed anhydride bond. You see the bond here, and another ATP provides the driving for the amide bond of carbamoyl phosphate. So, 2 molecules of ATPs are used in the first reaction. So, this is an energy dependent reaction. We have to make an expenditure. Now, N-acetylglutamate is an allosteric regulator, allosteric stimulator you can say. Without N-acetylglutamate, carbamoyl phosphate synthase I will not act at all. And this carbamoyl phosphate synthase is the rate limiting enzyme of urea cycle, which controls, and this is called a pacemaker enzyme. Note down one part, this is called a pacemaker enzyme. It paces the urea acid cycle. So, why it is called? Because carbamoyl phosphate synthetase is activated only in presence of N-acetylglutamate. In short form, it is called NAG. Now, this is an allosteric activator. Now, this NAG is produced and broken down. So, there is a balance of synthesis and breakdown. Now, only when this is available in adequate amount, then only it is available in the steady state concentration. Means their breakdown and synthesis is remaining same as per need of the cell, because this is an energy consuming procedure. We have to remember. So, basically steady state, note down, this one is not available perhaps in your book. Steady state level of energy is dictated, I repeat, dictated by the rate of synthesis and breakdown. Acetyl-CoA combines with L-glutamate, it forms N-acetylglutamate synthase. Just wrote down only this part, and other part if required I will tell you. Rest is all are available in the book. N-acetylglutamate and coenzyme A, in the next step acetylglutamate is broken down by hydrolysis to L-glutamate. So, once it is broken down, that means it will no longer act. Resynthesis will be required. So, steady state level of energy is required for urea cycle to operate. Okay. Now, next step, step 2. Now, this step carbamoyl phosphate synthesis has been covered. Now, carbamoyl second enzyme is ornithine. You just understand this thing, it is written in the book, you will get it. Ornithine transcarbamylase combines carbamoyl phosphate with ornithine to form citrulline. You see the amino acid which is present, first amino acid which is still there in the citrulline. This reaction takes place within the mitochondria. But you can see in the cycle, this ornithine is a substrate, not the product. The product is citrulline, that is shown in the cytosol, not in, you see here, carbamoyl phosphate and ornithine to citrulline, it is formed, citrulline is the product. So, there is a transporter basically in the mitochondrial membrane, that is an exchanger between citrulline and ornithine. So, one molecule of ornithine goes in and one molecule of citrulline goes out of mitochondrial membrane. That acts as a transporter. That is why ornithine is there inside and citrulline is there outside. Without that transporter, that disease, that cycle will not operate. So, this is called ornithine carrier isoform 1, ORC 1. It is a mitochondrial ornithine transporter 1 or ornithine citrulline exchanger, because it is exchanging. One molecule is entering, it is acting as an antiport. One is entering, other is coming out. It is a transmembrane carrier protein, mitochondrial inner membrane protein basically encoded by one gene. This gene was earlier known as ORNT, that is ornithine transfer 1 gene. Now, this name has been changed. This was discovered sometime in 1999. So, current name is ORC1 of this transporter. Step 3. Now, here you see citrulline was formed. From citrulline to argininosuccinate. Here you see ATP is required. ATP is getting converted into AMP, means ATP to ADP, ADP to AMP. That means 2 molecules of ATPs are basically consumed. 2 high energy phosphate bonds are actually required. Now, in this part you see the aspartic acid, which is having an amino group. This amino group is now coming into the argininosuccinate. So, one amino acid is produced by contributed by the ammonia, and the second amino acid is second nitrogen is contributed by aspartic acid. Now, this has been incorporated, that basically we want to produce double nitrogen in the urea, and that cycle, that structure we are trying to prepare through this reaction. So, in the first cycle we have used 2 ATPs, and in this cycle also there are 2 ATPs. So, 4 ATPs have been used. Now, argininosuccinate basically now gets converted into arginine through an enzyme called argininosuccinate lyase, and the aspartate which was there is getting after giving this ammonia is getting converted into fumarate. So, argininosuccinate, now these two amino nitrogen group is there. Now, argininosuccinate is basically broken down to by the argininosuccinate lyase into arginine. Okay. Now, this is the bond which needs to be cleaved. This was the bond which was cleaved to form this fumarate, and this now the bond needs to be cleaved here. The next step. So, this next step, this arginine was cleaved by arginase enzyme to produce urea and ornithine. Now, this ornithine which was required in the second step is now regenerated. The urea at the end we got, these two ammonia we got it. You get it? Two ammonia, two nitrogen, where from we got it? One is from ammonia, another is from aspartic acid. Now, the cycle will operate. You see here what urea cycle, what is happening here. So, these two steps are occurring within the mitochondria, less 3 steps are occurring in the cytosol. If you look at the whole reaction, our basic purpose is to get rid of ammonia and also carbon dioxide. So, if the basic purpose is to get rid of this ammonia and urea, that we have obtained it by forming used up as urea, that is the desired purpose of the cycle. Now, while this cycle is operating, aspartate which is having an amino group is losing its amino group and getting into fumarate. Now, if the cycle has to be operated, carbon dioxide will be available from electron transport chain, ammonia will be available from protein breakdown, amino acid breakdown. Now, all other things you see, they are remaining in the cycle. Urea will be produced, it will be drained. Only thing, aspartate will be required. If aspartate is not given, then the cycle will not operate. So, there will be deficiency of aspartate after each cycle. So, aspartate has to be regenerated somehow, and the whole reaction is occurring within the cytoplasm. Do you get my point? All the materials are basically are these we want to get rid of, we got rid of by making an urea. Water is available. Only biomolecule aspartate is getting used. Is this part clear? So, aspartate has to be supplied into the system. Now, how aspartate will come? Every time, every cycle will operate and every one molecule of aspartate will be consumed. So, if you recall TCA cycle, at the 7 and 8 step I have noted it down, this one, I have enlarged it here. You see fumarate is produced in this reaction, you see fumarate is produced from aspartate to fumarate. Do you get it? Now, this TCA cycle is operating in the mitochondria, but this reaction is occurring within the cytoplasm or cytosol, I would say, cytosol. This TCA cycle, it will not get TCA from the TCA cycle. So, there is an analogous reaction just like this one of TCA cycle which is occurring within the cytosol itself. So, you see here fumarate getting converted to malate to oxaloacetate, and while it is producing oxaloacetate, one molecule of NADH is produced also. The same reaction is occurring by the cytosolic fumarate and malate dehydrogenase, not mitochondrial. Now, two molecules here you see fumarate to malate and malate is getting converted into oxaloacetate in the same way, just like a TCA cycle. Now 2.5 more molecules of ATPs are generated here because NAD to NADH, and this NADH will be oxidized in the electron transport chain, and that will generate 2.5 moles of ATP. We have consumed already 4 molecules of ATP, now 2.5 molecules of ATP are generated. That is why the energy consumption total for this reaction is only 1.5 ATP. Now, oxaloacetate cannot participate. Yesterday I told you that oxaloacetate needs, there is a transamination reaction, and through a reaction called aspartate transaminase or glutamate oxaloacetate transaminase, this oxaloacetate will be converted into aspartate, and now it will act as a supplier of aspartate continuous to allow the cycle to operate. Do you get my point? We got produced, I mean the nature has evolved a mechanism by which aspartate is produced into the cytoplasm itself, where transamination reaction is occurring, and also at the same time getting NADH and getting 2.5 molecules of ATP. So somehow it is saving the energy. So at the end, when a urea cycle operates, the total energy consumption is only 1.5 ATP. 4 was used, 2.5 was generated. Okay. Now total energy expenditure per cycle is 1.5 ATP. Do you get it? Would you understand? Would you understand what I have said? Now, if you make a major change in the diet, like in prolonged starvation, there will be breakdown of protein. So, extra load of amino acid will be there. So, urea cycle enzymes have to handle that extra amount of ammonia into urea. So, urea cycle enzymes has to be increased manifold. It may be 10 to 20 times. Even if you take a large meal, large protein meal, I would say, then that extra amino acid will be produced. So, extra enzymes will be required. So, that will be increased. Now, urea cycle enzyme defects are having, there is 5 different types of enzymes are there. So, all these 5 enzymes are basically autosomal recessive. One enzyme you note down, it might come as an MCQ. Enzyme ornithine transcarbamylase is X-linked recessive. All other urea cycle enzymes are having a genetic defect of autosomal recessive nature, and one is only X-linked. You know the difference between the autosomal recessive and X-linked recessive. What does this mean? Have you got any idea? Okay, otherwise you have to take a genetic class on this thing, because without knowing the implication you will not know it. When the autosomal recessive means a patient will be affected only if 2 genes are obtained, defective genes are obtained from both the parents, then the disease will manifest. But in case of X-linked recessive disorder, males are affected in X-linked disorder because there is no 2 X chromosome. In females, they will be protected because there is an additional X chromosome, double X. Yes, males are in X-linked inheritance, usually males are sufferer. Females, they are carrier. And in this process, unfortunately, if a male baby is born, mother is blamed. Mother is blamed for everything, for giving birth to a girl child, and also, and if by chance, unfortunately, see, we are all living, there is every chance of getting an inheritance or mutation. So, if the mother inherits the gene, by chance, if her son gets affected, she will have no words to console herself. That is because of me only. So, that is a big ethical issue and psychological issue also. So, clinical features of all types of defects are almost similar, because either a given enzyme or a multiple enzyme catalyze the successive reactions. So, we have to detect whether the patient is suffering from this urea cycle defect or not. So, that is why newborn screenings are there. If required, I will take one more class and I will discuss something about this thing. So, once the baby is born, a battery of tests are done. Errors. By this time, you have known several enzyme defects. These are a very much rare condition. This does not occur. Normally, I have not also seen. Occasionally, you will come across with such type of disorder. But if a mass screening is made, perhaps some of the diseases can be detected. And that is done by the, right now it is done by mass tandem, mass spectrometry. Just note down this machine, name of the machine. And of course, if you do a DNA analysis, then you will be able to diagnose the type of enzyme, because the enzymes are encoded by the gene. So, that particular genetic study has to be done, DNA analysis, so that whether the defective gene is there or not, that we have to study. Okay. See, now I want to have an interaction with you. Since you are all limited students, you have to tell what type of enzyme is this one and what happens if the reaction, this enzyme is defective, means deficient. There is a possibility, I told you there are 5 reactions and 1 transporter. There's total 6 diseases will be produced from urea cycle defects. So, if carbamoyl phosphate synthesis 1, carbamoyl phosphate synthesis 1, you see 1 is here, 1 is defective. What type of enzyme is this? What type of reaction it is catalyzing? Do you remember enzyme classification? How many of you can tell enzyme classification? These are the things which will be asked during examination. They will not ask straight cut question. They will show the reaction and they will ask what type of enzyme it is. And at least the pattern of type of examination of what I am seeing in the, it is becoming increasingly difficult. Just orient you. What type of, you don't remember the enzyme classification. So, there is no question of, remembering. You can telling basically what type of reaction is catalyzed. So, if you don't read the useless class, can anybody tell about the enzyme classification? You just try, make some mistakes, I am there to help you. Have you read at all what is enzyme classification? No. Then I cannot basically teach you. I thought this will be a question and you will answer. The EC number is there. From the EC number you can detect it. If you are in a doubt what type of reaction enzyme it is, what type of reaction it is catalyzing, from the EC number you will get it. Now you have not read it. So, it is useless to discuss. So, this enzyme is basically, see there are 6 different types, in some books there are 7 types. First one is oxidoreductase, second one is transferase, third one is hydrolase, fourth one is lyase, fifth one is, jore bola, fifth one is, ঠিক করে বলো, just tell correctly, isomerase, and sixth one is ligase, okay, and seventh one is translocase, okay. Now here what type of reaction it is? Ligase. Why do you say ligase? If you are in a doubt at all for any reaction, I have emphasized, look at the EC number. EC number is ligase. So, what is it ligating? It is ligating carbon dioxide and ammonia, isn't it? So, this is this EC number, and if you block it, if the enzyme is deficient, then the ammonia will not be utilized, not be produced to form carbamoyl phosphate. So, there will be a hyperammonemia. Ammonia level will increase in the blood because it cannot proceed further. So, that is called a type 1 hyperammonemia. Ammonia level will increase in the blood, so that will cause hyperammonemia, and if it is a defect in the carbamoyl phosphate synthesis 1, that means it is hyperammonemia type 1. Okay. Second one, what is this carbamoyl phosphate? Again conjugating with ornithine to form citrulline. What type of enzyme it is? Yesterday I told you several times about transaminase. It is transferase, basically transferring the transcarbamylases. Carbamoyl group is getting transferred. So, this is EC number 2, and if you block it, then there will be hyperammonemia as well as uric aciduria. Because you see here in the first reaction, the carbamoyl phosphate is there. In the second reaction, block is here. So, carbamoyl phosphate will also be deposited, will be in excess, because the reaction has proceeded up to this stage and there is a block. And this carbamoyl phosphate will lead to increased pyrimidine synthesis, and that pyrimidine synthesis will lead to uric aciduria. So, uric aciduria will be an additional feature. So, hyperammonemia plus uric aciduria, if you get, the block is at the level of carbamoyl ornithine transcarbamylase. Okay. Now, this is a transporter defect. This transporter, if it is defective, then it will cause a HHH syndrome. That is, there will be hyperammonemia, there will be hyperornithinemia. You see here, ornithine cannot be transferred. So, there will be a hyperornithinemia, and citrulline will also be affected because citrulline cannot be produced. So, hyperammonemia will be there, hyperornithemia will be there, and in addition homocitrullinemia will be, urea will be there. Why homocitrullinemia? Citrulline we have not said. Because of excess amount of carbamoyl phosphate, you see here ornithine is not able to enter into the mitochondrial matrix. Instead, lysine will enter, and that will react with carbamoyl phosphate. For this is a modified version of lysine. So, lysine will combine with carbamoyl phosphate to form homocitrulline urea. You note down this part. This may not be available. Why homocitrullinemia occurs or homocitrullinemia occurs in HHH syndrome? Because carbamoyl phosphate ornithine cannot enter, instead lysine will enter into the mitochondrial matrix and it combine with carbamoyl phosphate to form L-lysine. Okay. So, somebody of your class wanted a review where from we get it. So, the source of this thing is from Molecular Genetic Metabolism 2014, that student is not available. They wanted where from we are getting all these things, research articles. So, everything is not, this part is not written in your book. So, I had to collect it from the journal. So, again this is from Genetic Review, which is published online in 2025. This, what is the actual, where from it is coming. So, it was coined by the term by Vivian C. in 69. At that time, he did not know all these things. He knew only ORNT1, that is ornithine transporter, was invented in the year 1999. But he knew that there is a defect in the ornithine transport, but it is due to ornithine transport that was not known to him. And he described this disease. So everything what somebody sees may not be explained every time. See, you see there is a time lag of 30 years before this thing was discovered. But he made a good observation. Step 3 is argininosuccinate synthesis. Now, if this is blocked, what type of again, you see what type of enzyme it is? Citrulline and aspartate, this is combining. So, that is a ligation basically. So, that will be a ligase enzyme. That is okay. So, that is why EC number is 6.3, and this will cause, because block is here. So, citrulline, up to this citrulline urea cycle will proceed. That is why there will be citrullinemia or citrullinuria. It cannot proceed further. So, any block is there, the metabolic products which is proximal to it, before it, will get accumulated in excess amount and that will get excreted. That is general principle. You have reached some part, but then after that part you are not able to go. So, you have reached up to that much distance, or that much reaction have been covered. So, that much metabolites are available, and that will be in excess amount because it is not getting path. So, there will be some regulatory mechanism to stop it, or there may not be any regulatory mechanism, so excess amount will be produced, and that will give a clue to the diagnosis. Otherwise, you will not be able to diagnose where is the defect. Now, step 4 is argininosuccinate lyase. You see what type of enzyme? Lyase is there, already name is there. So, under what classification? What will be the EC number? EC number? Bolo jore? 4, yes, 4. Hydrolysis is 3 and lyase is 4, because lyase does not require water. So, here there is no water requirement. Do you get it? So, this is basically 4, correct you have said, that is called arginosuccinic aciduria, because arginosuccinate will not be able to get converted. So, this will get accumulated. So, this will cause arginosuccinic aciduria. Okay. So, this is how you have to remember, because otherwise if you do not remember the cycle, you will not understand, and we will ask this question. Step 5, arginase. Arginase is getting, from arginase to ornithine. What type of reaction it is? You tell something, at least one student is responding, 2 students are one student is attempting. Just tell something, you will be able to tell. Huh? Jore bolo? Hydrolytic. Yes, correct. Because water is required here. Dekho keno chhela, why somebody gets a good mark or why somebody other don't get it. Because in this year, last year, one examiner came, he gave marks 20 out of 20. Only asked one question, he answered correctly. A graph was asked to be made by him. He immediately did it. And 20 out of 20. That indicates that you know it. So, yes, it is 3.5. This is called arginemia, because arginine will be present in excess amount because block is here. Okay. So, clinical features of urea cycle disorder will be basically of neurological in nature. But when the baby will be born, it will be very difficult to diagnose because they cannot say anything. So, there will be some neurological manifestation like vomiting, abstinence of high protein food, Because see, a baby will take only mother's milk, but he or she may not want to take milk for some many other reasons. It is not that urea cycle defect. You see, intermittent ataxia will occur if the baby can walk. That newborn baby cannot walk. So, unsteady gait you will not be able to know. Lethargy, it is not possible in a newborn baby. Severe mental retardation not possible to detect at all. These are the features. When the baby grows, then only you will be able to detect, but at the time of birth it cannot be. But if you screen it, the newborn screening, where it is done basically by a heel prick, blood is taken and that blood is sent, filter paper blood is sent to a higher center for detection of these diseases. Now, because of the tandem mass spectrometry, a large number of metabolites can be detected at a time and within a short period of time, and that was not there during our time. So, there will be characterized, ammonia will be there, provided it is occurring in the first and second step. I repeat, because at that time only one ammonia has been trapped, and then argininosuccinate, 2 ammonia have been trapped. So, the hyperammonemia effects will not be there if the disease is beyond argininosuccinate. Do you get my point? Ammonia will be utilized in 2 reactions, in the first reaction itself. So, 1 ammonia is consumed. So, effect of ammonia will be there definitely, but when 2 ammonia has been consumed in the argininosuccinate stage, so there will be no ammonia, the hyperammonemia effects will not be there. So, it is intoxication is most severe when the metabolic block is at reaction 1 and 2, not in others. And 4 out of the 5 metabolic diseases results in the precursors of urea. Urea is the last step. If there is a block, there are 5 reactions I told you. So, out of this is the last reaction was the urea formation. So, if that step is blocked, last step, so precursor of urea will be available in the blood. So, treatment, what will you do in urea cycle? Listen, first and foremost you have to diagnose the case, otherwise you cannot treat. The baby requires protein, the mother's milk is the only food what the baby can take. We can tell many things, but it is difficult to implement. So, here low protein diet, normally the baby takes the milk from the mother's breast. Now, that amount has to be restricted. You have to take the baby out from the breast, so that small protein is allowed at a time, so that the small amount of ammonia will be there, so there will be less ammonia intoxication. That is the only thing you can do, and arginine and energy can be given, needs to be given orally also by some means to promote growth and development, and basic purpose is to avoid metabolic consequences of this thing, but it is very difficult and most of the baby do not survive also because of the defect. Now, you see, when the baby is born with defect, then the mother has to accept he cannot kill that baby. It is difficult. That is why I took up a project as early as late 90s that I will develop a center at Durgapur Steel Plant Hospital for the Eastern India, which is not in sight at all, even in 2025. That if the unborn baby, when the baby is in the mother's womb, if at this stage the defect can be, disease can be detected, that the baby is going to be born with a genetic defect, the mother will be given an option to get the baby aborted, and all mothers will want it. So, that study has to be done. So, by this time, you know when the baby is conceived, the fetal tissue is there. Now, we have to, at that time, we have to take either a fetal tissue or you have to take the chorionic amniotic, the fluid on which the fetus moves. Now you have to put a needle. This is dangerous procedure, because that will cause abortion itself. And I need an ultrasound specialist. I need a gynecologist also. So anyway, I conceptualized the first prenatal DNA diagnosis center for Eastern India at Durgapur Children's Hospital in May 2022. This is for the unborn child. And I submitted a project to the Ministry of Biotechnology. Biotechnology in 2002. I mean, as you can see, so this project, ambitious project of setting up the Professor will be knowing, much better. This is still not in sight, even today in West Bengal. But I wanted to make it in for the Eastern India. And SGPJ Lucknow, SBJ is a postdoctoral institute, means where the DM and MCH is taught. I went there, I got myself trained up, and I had to motivate the director, because they wanted to set up an off-campus center for the first time from the SGPJ to Durgapur, that this will cater to the Durgapur area. This was a very difficult task for me, because I had to convince at this stage, when I was only a 11 years post-PG candidate, and the 10 candidates interviewed me like anything, and the project was cleared, and they said that you are competent enough to do this thing. So I was the principal investigator. The project was submitted. And this is the project. This was again, since this will be in Durgapur Children's Hospital, so we had to get a clearance from Ministry of Steel from New Delhi. So that was a massive effort, because this hospital was dedicated for Durgapur Children's employees. But we will extend the facility to other people. So the clearance was there. Director also signed it. It was published with much fanfare at Lucknow edition of Hindustan Times, both in Bengali, English and Bengali version on 7th May 2002. And I was really having a great delight that I am going to have the project. But my greatest crime which I made, I did not include HOD. And because of red tapeism, this project was not sanctioned. So the dream remained unfulfilled. Some of you can take it up and simply do it. And I have attempted many things, out of which this is one. At the end, I published a paper in the Indian Journal of Human Genetics. It is very difficult to publish a paper or review article in an Indian journal from a general hospital. So when I was a very young person, it was published in the year 2004, and this is my article, where I did not want to do the test of prenatal DNA diagnostic from the mother's womb, that is either from the chorionic villus or from the Lycaramni, but from maternal blood, because in the maternal blood, as early as 5 weeks of pregnancy, fetal DNA can be detected. So even the mother becomes aware that she is pregnant, that sex of the child can be determined using mother's blood, and that technology was known to me when you people were not born. And ultimately I had to satisfy myself, giving a patting on my back, that this is the project which I wanted to do it, but the love's never lost.